Hi everyone, we already know, so we'll make a start with this. Welcome to our Alvetex webinar. This is session 1 of our beginner's guide on evaluating 3D cell cultures. Your hosts for today are myself, Fred, as well as Ross and Dan. I specialize in imaging, Ross is our expert chemist, and Dan takes care of liver applications and protein science. We all work in research and development here at reinnervate, and we also double up as your tech support team. You can contact us at any time using our individual emails, which you can see on this slide, but also using the general tech support email, our phone number, it's a UK number, or by filling in the contact form, which you can find from our website. And we are all here today to help you accelerate your success with your Alvetex samples. We will be covering quite a few subjects in this seminar today, and I am going to start with getting you to think a bit more about why you should be using your 3D culture in your research. Quite simply, 3D culture can give you more representative data which will result in more relevant research. You can develop complex models such as co-culture or invasion models, 3D cancer models for clinically relevant drug testing, and generally you can increase the translational value of your research. Basically, you will be generating better, more relevant data and publications. If you are wondering if anyone else has already used Alvetex 3D technology, well yes, Alvetex is currently being tested in many countries worldwide, and to the right on this slide you can see a few examples of institutions where Alvetex research is being carried on at the moment. People who have used Alvetex are certainly happy with it, and you can see a quote at the bottom of this slide. If you want to read more testimonials for yourself, you can have a look through the web link which is at the bottom of the page. Now I will be handing you over to Ross, who is going to tell you a bit more about the chemical structure of Alvetex and how it can help you achieve genuine 3D cell culture.
Thanks, Fred. So what is Alvetex technology? Alvetex is a highly porous scaffold. It's around 90% porosity, in fact. It's made of inert, cross-linked polystyrene created using an emulsion templating technique. This templating technique allows for routine and reproducible control of the Alvetex structure, its mechanical properties and porosity. The Alvetex structure consists of voids, which are the larger circular structures which you can observe on the micrograph images in front of you. Each void is approximately 40 micrometers in diameter. The voids are connected to one another by interconnects or pores, which are approximately 13 micrometers in diameter. The monolithic material produced can be engineered into membranes and sheets which are 200 micrometers thick, as shown in the top right-hand image. So what Alvetex actually does, it introduces a third dimension into which cells can grow and occupy. It really introduces space effectively, that 90% porosity as I mentioned in my previous slide. So if you look at the top line, this is a situation in conventional two-dimensional culture. These are the vessels that are classically used, and of course it is only two-dimensional, therefore the cells grow as a simple monolayer, as you can see here. Alvetex introduces this Z dimension through its scaffold. It provides great support and vertical space into which cells can occupy, and they pile up on top of one another, ultimately creating three-dimensional tissue constructs. Cells are able to adapt to their normal morphology and form numerous cell-cell contacts and organize into their natural in vivo structures. So Alvetex delivers genuine three-dimensional cell culture. Our Alvetex technology supplies sterile by gamma radiation until removed from its packaging. As discussed in my previous slide, Alvetex is made of cross-linked polystyrene which is hydrophobic. This means it is non-wettable to culture media. The Alvetex disc can be rendered wettable by washing, hydrating with 70% ethanol prior to cell culture. Dan and Fred will discuss this later on as they lead you through the protocols and workflow sections of the website. Our Alvetex is available in a range of different formats that you, the customers, already use, including well plates, well inserts, and for longer term cultures, our well insert holder in deep Petri dish. All this information regarding our wide-ranging product formats can be found at www.reinnervate.com/Alvetex/formats. So the key things to remember about Alvetex technology is that it delivers genuine three-dimensional cell culture. It's very simple and easy to use, and it's supported by a large number of protocols on our website, as you'll soon find out. Each product format is made from the same inert polystyrene scaffold with routine control over structure and porosity. There is also no need for any changes to your downstream analysis. So I'm now going to pass you back to Fred, who's going to go through the next section of the webinar. Thank you for listening.
Thanks, Ross. Now, let's get you started with those samples. We realize that when you start your evaluation, you will have a lot of questions, from how many cells do I have to seed onto one scaffold, and how can I see my cells through the scaffold, all the way to which assays are compatible with Alvetex, and can I extract RNA, DNA or protein back from my scaffold. The thing to remember is that Alvetex is a very simple system to use. We have loads of protocols available online, and Dan will be talking to you about these extensively in a few minutes. And you don't need any new or special medium or equipment, or even expert training to use Alvetex. What you do need to do before you start your experiment is to check out the online protocol workflow. You can see the direct web link here. You can also get to the workflow by going to the home page and then clicking on Tech Support and then Alvetex. This online protocol workflow will direct you to the default page, which is this one, Preparation. Once you're there, just stay there. You can see that the first protocol which is listed is called the Quick Start protocol. In the Quick Start protocol, you will find a lot of general information on the early stages of your Alvetex experiment. Things like what to remember before you start, precautions when handling the samples, guidelines on cell seeding, and on which volume of medium to add. And for insert formats, we also have guidelines for feeding options. Here is an example of the kind of data you will find in the Quick Start protocol. Dan will go through this in more details in a little while, but just for now, we'll just have a quick look. You can find ranges of recommended cell densities, as well as feeding volumes, and this for all of our formats. Another very useful section of our website is called the 3D Learning Zone. This is where we keep an archive of all our educational videos, and when you're starting, the most relevant videos are really these three at the bottom of the page. This will show you, rather than just tell you, how to handle your samples and how do you fit the inserts into the culture plate, how to pre-treat your Alvetex with ethanol and medium washes. Remember, Alvetex is hydrophobic when supplied, so you do need to do your ethanol pre-treatment before use. And finally, this video will show you the exact technique for cell seeding, and this for all of our formats. Now I will hand you over to Dan, who is going to show you in details how to navigate through the workflow.
Okay, thank you, Fred. Hello, everybody. My name is Dan Maltman, and what I'd like to do over the next 5 or 10 minutes, if I may, is lead you somewhat briefly through our information and protocols workflow online. There is a large amount of information in that workflow, and so an exhaustive tour would take a very long time. What I'll do instead is open up each section and show you how to access the materials, and also illustrate at the same time with some example data. As you can see from the list there, the workflow is subdivided into 4 main sections: preparation, setup, monitor and analyse. And these 4 sections cover different aspects, from preparation, getting it ready for cell culture, right through to performing specific analyses at various points within your experiment. So the first section, as I said, is called preparation, and if we move into this window on the website, this is what we'll see. And in here you'll find important information which you'll need to be aware of before you get started. You'll also see access to frequently asked questions and answers, and also contact details for technical support. And as mentioned by Fred a few moments ago, the most important go-to document in this section really is the Quick Start protocol, and we'll take a little look inside that now. And the protocol here provides basic information on how the scaffold should be handled and prepared to get it into a ready state to receive cells. But more importantly, some key information as to guidelines for cell seeding densities. And this is what you can see in this table here. And I should point out that this is guidance only based on our own experience with a range of cell lines in our laboratories. And we usually recommend that the user performs a degree of optimization themselves, particularly if they're using a cell type which isn't covered in our cell-specific protocols, which I will discuss in a few moments. A second table within the Quick Start protocol, which is also very important, deals with the recommended volumes of culture medium for the different formats. And what you'll notice with the well insert formats is there are a range of different volumes there, and that really reflects the versatility of the format, in that you can raise the level of the medium in the outer chamber to the point where you can have feeding from both above and below the membrane, or from just below. And in the latter there you would find utility, for example, in an air-liquid interface system. And as Fred also mentioned, the preparation section also contains some very useful instructional videos. This is just a still from the preparation and handling videos. And what these provide really is a visual instruction on how to actually physically perform specific steps during correct Alvetex use. And as you know, it can sometimes really help to see how it's actually done rather than just read about something. So if you have the time, we'd really recommend you take a look at these. They're not long, and they contain a lot of useful tips. So moving now into the setup section, once you've understood how to prepare the scaffold to receive cells, you'll be ready to actually start preparing the cells themselves and getting ready to seed your cultures. And so this is an invaluable section. And of particular interest, I would say, are the cell-specific protocols. And also there are a collection of optional protocols for pre-treatment of the scaffold with extracellular matrix type products and reagents. And so we can click on the cell-specific protocols icon, and what we come up with is a list of all the cell types for which we ourselves have performed validation and growth conditions optimization within our laboratories. And you can see that a wide range of tissue types and disease states is represented, and there are also some examples of primary cell types as well in there. In terms of what these protocols contain, apart from specific details on how to prepare your cells, what sort of densities you ought to be seeding, you will also find representative data in the form of histological images, and also some cell viability data as well. And the example image which we've pulled together here just provides histological data from 4 different cell lines or cell type scenarios. And of particular interest, if you look at the bottom right hand image, that's actually a co-culture of dermal fibroblasts within the scaffold, and they're supporting the growth on top of some keratinocyte cells, so in essence a full thickness skin model. And as I mentioned, there is also a collection of coating protocols within the setup section, and these are very useful, especially if you think that your cell type will require the presence of an ECN reagent before your cells are introduced. The 2 examples depicted are both based on collagen 1, but they're both very different. The left-hand image, it shows cell growth patterns of HepG2 cells seeded onto scaffold, either treated or untreated, with a very thin, non-gelling layer of collagen 1. And what we find with this cell type is that a coating such as this will increase the rate of penetration of cells into the scaffold. Whereas on the right hand side we still have a collagen 1 treatment, but in this case it's been presented in a gelling solution, resulting in a very thin and intact layer of collagen gel right across the surface of an Alvetex scaffold disc. And there's a nice SEM image as well at the bottom, showing a nice sheet of collagen. And this type of application will be particularly useful, for example, if you were trying to establish a 3-dimensional monolayer of epithelial cells. And now moving on to our monitor section, this is where we start to provide some actual analytical methods. And as the name suggests, these methods are intended to allow you to monitor the progress of your cells throughout your culture duration. And broadly speaking, the protocols contained within the monitor section deal with cell metabolic assays, viability assays, and also some microscopic techniques for cell visualization. So taking the metabolic protocols, we can click on the icon and see that we come up with a list of assays there. At present there are 4 different assays listed. We would anticipate that this list will grow as we test different kits and different reagents. So for example, we already know that XTT assays and commercial kits such as ProMega's cell titer glow assays all work very well with Alvetex scaffold. We've just presented there some example data from an Alamar Blue and an MTT viability assay, and also a Pico Green quantitative assay for extractable DNA. And what these assays will allow you to do is compare viability from one state to another, or alternatively to just track the progress and the rate of cell proliferation within your own particular culture setup under your own growth conditions. So very useful methods for tracking, monitoring the progress and the health status of your culture. And the alternative means of following the progress and the confluency of your cells and potentially the health state of the cells within an Alvetex culture is to look at them via microscopy. And of course it's important to realise at this point that when you make that transition from a standard 2D monolayer into any 3D system, there will be challenges faced in terms of full visualisation of all the cells within that culture. So in any 3D culture system, it's going to be difficult to clearly focus on all of the cells at the same time. But what we find, for example, with Alvetex is that if we implement a light staining method, such as the use of a neutral red dye, then the scaffold can be mounted and viewed very clearly under a light microscope, giving a nice indication of cell morphology, cell health, and also the confluency, the thickness of growth across the surface of the material. And then for more sophisticated and complex methods of microscopic analysis, we can use confocal imaging, either as an endpoint visualization or to monitor the growth of live cells, as you can see there in the image on the right. And the final section of the workflow is entitled Analyze, and what we provide in this section is a collection of analytical methods to assess the biology that is taking place within your cultures. The section includes methods for visualizing cells, recovering cells for downstream analysis, and also extracting nucleic acids and proteins. So in terms of cell visualization, if you're prepared to fix and embed your samples, you can come up with some lovely pictures, very clear, high-quality images of cells in the scaffold. This slide has some example data from different histological methods, and what you can appreciate is that most common histological methods are compatible with the scaffold. On the left, we see some immunostaining of the proliferative marker Ki67 in wax-embedded Hackett cells, and also a nuclear counterstain, all taken from within the same section. And the middle image is a histological stain, it's hematoxylin and eosin of some wax-embedded HepG2 cells. And I think what this image shows, and I hope you'll agree, is some lovely 3D cell morphology, healthy cells, and some extensive cell-cell interactions taking place. And finally, the image on the right is a tall blue stain of some resin-embedded Hackett cells, showing some really fine detail of cellular structure. And the last content from the Analyze section I'll touch on is just the fact that you can very readily isolate protein and RNA or DNA from the scaffold, and this can be performed in situ without taking the cells out first. So the cells can be lysed within the scaffold, and these molecules can be extracted using the appropriate kits or reagents. We can just see on the left an example of some protein yield data with SDS page profiles and also a Western blot. And on the right, some RNA extracted and analysed by agarose gel electrophoresis. So I think, as I mentioned at the start, there's an awful lot of content within the workflow, and you can find it and access it easily the way I've just shown. And if you want to consult a single document, then the compiled protocol document contains all of the information in one place, and that document can also be accessed via the workflow. And if I may, I'd like to hand back to Fred, who's going to touch on a few small points before opening the session up for some questions and answers. Thank you.
Thanks, Dan. If you are interested in more complex culture systems than what is offered in the protocols, or if you want to see more example data, more complex data sets, we do have a page on our website which is dedicated to what we call application notes. These are in-depth studies which will show you the benefits of using Alvetex in specific research areas, and you can see the research areas that we have data for at the moment on this slide. You can see there is quite a variety of them. These include detailed protocols and a lot of data, and these data are quite varied, and they will show you what morphology and cell response you can expect from your cells when you grow them in Alvetex. Alright, now I will just introduce you to our competition that we are running at the moment. It is called the Alvetex Evaluation competition. Some of you might have received an email about it already, but if you haven't, don't worry, here are the rules. We have 3 prizes of £500 or the equivalent in US dollars or euros to give away, and these should go toward the attendance to a conference of your own choice. You can note as well that the first 30 entries will get an Amazon voucher, so it is worth entering this competition and entering it quickly. All you need to do is submit an abstract with one figure, and we are preferably looking for something which has immediate visual impact. The deadline is on the Tuesday 30th of April, and the winners will be announced sometime in May. If you have not been sent an entry form, as I mentioned before, you can just get one by emailing me at the address which is shown here. We have now reached the end of the seminar, so we are going to open for the question and answer session. Myself, Ross and Dan are all available to answer your questions, so do feel free to go and ask away, and thank you for listening. Okay, we have a question. I will answer the first question. We have just been asked whether there is a direct way to access all this information that we've presented you from our homepage. Well, yes, in this webinar we have made a point of showing you the direct links to all our resources, but you can access these quite easily from the homepage. Basically, when you get to our homepage, which is reinnervate.com, you can see it on this slide, this is addressed to our homepage. You will see a banner at the top of the page, and it has got moving pictures. Now, this banner does have a tab, and if you click onto the Evidence tab, and then you click again on the banner, that's just the way it works, you will be directed to a hub page. We call it the hub. You will find here that you get the links to every single page you need. For the video page, you need to click to the 3D Learning Zone. If you want the application notes, it's there. If you want the protocols, it's there. If you want to see this webinar again, or actually any of the webinars that we have going, you can just click on live webinars. It does say live, but it's actually an archive as well. So you can access everything from this page, and hopefully that should be everything you need in there. Thank you. I will have a look. We'll select the next question. All right, Ross is going to take the next question.
Yes, hi. Somebody's just asked if Alvetex is chemically resistant. The answer to that is yes, it is, due to the actual cross-linked nature of the polystyrene, as I discussed in the presentation. But people must be aware that any organic solvents used, or any good organic solvents of polystyrene, generally will swell the actual material itself. It will just change in size. So you have to be aware that the surface properties of the material can also be changed if using any chemicals as well. Things like sulfuric acid can make the material more hydrophilic, etc. So if you have any problems with any of the chemicals that you're using, please contact our technical support team. Thank you.
I'll take this one, Ross. Is it possible to grow endothelial cells in Alvetex? Good question. As far as I'm aware, yes, we've experience of the growth of Alvetex cells, and also, correct me if I'm wrong, Fred, microvascular endothelial cells with hepatocytes. I believe that's true.
Yes, we have indeed. Yes, correct.
And I think there's evidence of that actually in our co-culture application note on the website. With cells like endothelial cells and also epithelial cells, they can be grown within the scaffold, but if you want to achieve a monolayer situation, then you would be recommended to apply a collagen gel, as in the slide that I showed earlier. And you can find details for all these methods on the website. Okay, thanks. There's another one, hang on a minute. Okay, so which kind of RNA and protein extraction kits are suitable? And a wide range is the answer. We've got some experience with the use of chloroform phenol methods for RNA extraction. There is no problem there with chemical incompatibility with the membrane. In terms of protein extraction, in principle any common protein detergent-based extraction reagent is fine. We'd be happy to give more detailed information via technical support, but yeah, certainly anticipate no problem with common reagents.
Okay, I will take this next one. We have somebody who asked whether it's necessary to section the samples to see the cell. It's quite a common question. We do have a lot of histology slide and section slides on our website, and this is because we tend to treat Alvetex cultures in the same way that you would treat a piece of tissue. If you think about your cells growing into Alvetex, they're going to multilayer, they're going to be 3D. Depending on your cell type, they can be quite dense. So actually putting them in wax and sectioning is a very good way to assess how deep they are going, and it's also a very good way to look at morphology. And you can certainly do some immunofluorescence if you're using antigen retrieval techniques and so on. But you don't have to. That's not the only way to look at your cells. As Dan touched on while he was talking you through the protocols, you can do confocal. That works very well for immunofluorescence. And if all you're after is looking at morphology and confluency of culture, and people do worry about that because yes, it is opaque and they worry they're not going to be able to see the cells while the cells are growing. You can use your neutral red staining. We do have a protocol, probably in the preparation part of the workflow, and you can use that with light microscopy, so it's not a complicated technique to use. We do have actually a white paper on imaging techniques, all the imaging techniques compatible with Alvetex, so you can have a look at that. That will be, I believe, in the monitor and should be also in the analyze section of the workflow. And we are preparing a confocal user guide as well, which is not available yet through the website, but if you can just email us at tech support, and we can send you a copy. So you don't have to section your cells, basically. It will give you really, really good results, but if you don't have the equipment, you don't have to do it. You can get around and still get really, really good pictures. Okay. All right, it's getting a little bit late now, isn't it? 5:30. So we've got to close the session, but don't worry for all the other questions which we haven't had the time to answer live. We will get back to you through tech support, so don't worry if your question hasn't been addressed. We will be taking care of this. We hope you've enjoyed this webinar, and don't forget you can go back to our homepage and navigate through to find the webinar again. It will be recorded and available to view again, possibly in the next few days. So thank you very much, and bye.